Hello, my name is Nistha Panda, and I'm a master's student at the University of Wisconsin-Madison. Today, I will be presenting our poster on prioritizing genetic variants in long QT syndrome using network analysis. Though there are many computational methods used to score or prioritize causal variants for rare disease, most algorithms do not look at variants in larger cohorts of subjects. We are interested in using network-based methods to identify novel variants and their relationships to known causal genes for subjects in a large rare disease cohort. We use genomic data from the NIH Olivos research program because it allows us to group subjects based on phenotype or disease diagnosis. Specifically, we look at long QT syndrome, a rare autosomal dominant disorder characterized by abnormal cardiac repolarization, which looks like a prolonged QT interval on an electrocardiogram. Deleterious variants in KCNQ1, KCNH2, SCN5, and other closely related genes can be involved in prolonging the QT interval. We find interpretable subnetwork solutions constructed with the prize-collecting Steiner forest algorithm that can be used to implicate causal variants and explore new candidate genes associated with QT syndrome. If we start off with a protein-protein interaction network and then connect subject nodes by adding outgoing edges to genes that harbor subject variants, we can assign a positive prize or a score to nodes of interest, such as subject nodes or genes with a known association to long QT syndrome. Then we can use the prize collecting Steiner forest algorithm to traverse the network and find a subnetwork that connects the nodes with a positive price while minimizing the cost of adding more edges. To do this, we start off by defining a set of candidate genes per subject. We use SNP and indel exome variants for around 1,200 subjects that have a OMOP concept ID for long QT syndrome. We then filter their variants on molecular consequence, Rebel scores, and allele frequency, and aggregate on the gene to get a set of candidate genes for each subject. We map prizes to a background string protein-protein interaction network. We add a uniform positive prize to subject nodes and a set of 23 genes that are associated with long QT syndrome and prolonged QT interval. And then we also add a small length-based negative prize to larger genes. Larger genes accumulate more variants, so we add the small length-based negative prize as a soft preference against choosing pathways with larger genes over smaller genes. We then use Omics Integrator 1 to map our input prize data to the protein-protein interaction network and run the prize collecting Steiner forest algorithm. PCSF minimizes the following objective function, and the subnetwork solutions are controlled by three hyperparameters: beta, which balances the node prices; omega, which controls the number of connected components; and mu, which assigns a degree-based penalty to control the number of high-degree nodes in the subnetwork solution. We use this signaling pathway reconstruction analysis streamliner, or SPRAS, to run Omics Integrator 1 with many different hyperparameter combinations to get a large set of subnetwork solutions. We then prune the hyperparameter combinations to get a plausible set of plausible subnetwork solutions that are relevant to the input price data that we can use for our final analysis. We use the pruned hyperparameter settings to get our plausible networks and combine those plausible networks to create ensemble networks to get node and edge frequencies. We use ensemble networks so that we don't overinterpret a single solution in cases where there might be multiple different good subnetwork solutions, and also to determine the stability of certain network features. We also create ensemble networks for a control cohort that is matched on sex, age, and predicted ancestry. We use the control ensemble networks as a point of comparison and to help explain away any inferred nodes that might appear because of network biases. Now that we have our ensemble networks, we can use them to prioritize candidate genes for each subject by assessing the connectivity of the candidate genes to known causal genes and the node frequency of the candidate genes in the ensemble networks. After filtering the ensemble networks for case subjects on edge frequency, we find three subgraphs that connect many of the genes that have a known association to long QT syndrome. We can look more closely at the candidate genes and subjects in these three subgraphs and how they are connected to genes that um, have a known association to LON-QT syndrome. We also perform a gene ontology enrichment analysis for high frequency nodes in our ensemble networks. Um, First, we select a set of Go terms that are relevant to prolonged QT interval and cardiac repolarization. Then we perform the enrichment analysis for high frequency nodes in the case and control networks, excluding genes that already have a known relationship to long QT syndrome. We checked if the enriched terms for each network include any of the Go terms that are relevant to long QT syndrome, or if enriched terms are descendants of relevant terms in the Go hierarchy. We see that high frequency nodes in the case networks seem to have more enriched terms that are relevant to prolonged QT interval and cardiac repolarization. We also can prioritize variants based on the node frequency in the candidate genes. For each subject, we rank their candidate genes using node frequency in the ensemble network, and we can look more closely at the variants in highly ranked genes. With that, I'd like to thank you all for listening to my presentation. This work is funded by the UW-Madison Research Forward Funding. We would also like to greatly acknowledge all of us participants for their contributions and the NIH All of Us research program for making this data available.